we are doing with Brachydontis pharaonis is a very easy study case, because this is an invasive species, and where he lives in Sicily is a very small pond, and now I'll arrive to it. Brachydontis pharaonis is a small bivalve, about 3 centimeters, is a Lesepsian species. Probably most of you know this definition, this term, no? Okay, your Mediterranean colleagues probably are more familiar, because these are Lesepsian are all species coming from the Indo-Pacific area, just entering through the Suez Canal, entering the Mediterranean and colonize the whole Mediterranean. Sicily is just in the middle of the Mediterranean, and divides the eastern from the western Mediterranean, and apart from the main to be a center of a lot of business, is also a hotspot of diversity and biodiversity, and a lot of invasive species just stop here, like, for example, these animals. About 400 species in just 100 years entering to the Mediterranean through the Suez Canal, arrived just in Suez Canal. It arrived just in Sicily and is colonized 20 years ago, just a pond here in the western Sicily. This is a small pond, 6 hectares, and then is partially closed, and then we can assume that immigration and emigration are not so important. We can assume also that dispersal is not so important, and we'll see. And before to apply this framework, we need to set the carrying capacity. The carrying capacity is the maximum, or the theoretical maximum number that one habitat is able to sustain of a certain the number of a certain species. And then in only 6 hectares of pond is easy through all is easy to measure all, account all animals inside, just applying the experimental design, just using photos and pictures, and all is possible just to understand which is the highest number possible in sustaining that this area is able to sustain as a function of the physical components of the pond and the amount of food in the pond, in this case as a function of the particulate organic matter available. We collected a lot of animals, we counted a lot of animals in just 3 years using quadrats, we measured a lot of them, we calculated, estimated just the cohorts and the distribution, the population structure, and then we depicted also the von Bertalanffy curves. We measured the age of animals, not all, just samples. The fact that this pond is semi-closed is allows us just to set the dispersal to 0, the larval mortality in these animals from literature is usually more than 99, usually 99.99. The immigration emigration is set to 0. The adult mortality by predation was measured through different caging experiments like Gray and Nova showed yesterday. Also, we want to use this framework, then we want to capture the environmental change variability in the habitat, translate it as a way to function from the side of the organism, and then we want to estimate the role of this environmental change on the number of eggs. And in this but we want also to try to get information about biodiversity. We can use, in this case, the concept of ecosystem engineer or the concept of species driving biodiversity. In this case, we went around Sicily in those six locations where Brachydontes is present, and we choose the 18 sites. In every site, we chosen 3 plots. In every plot, we had 3 quadrats, 20 by 20 centimeters. We went about 2 times per year, and then we have also the temporal component, and then we got the Brachydontes pharaonis, and then we measured all species associated to Brachydontes pharaonis, both epibionts and in the crevices in the Brachydontes pharaonis, and from natural and artificial substrata, and from both horizontal and vertical, because we want to use the relationship between density of Brachydontes in x-axis and the diversity in the y-axis, just to understand if we can infer, using this framework, on the effect of this the potential loss of this ecosystem due to environmental change of this ecosystem engineer just on biodiversity. And then we ran some scenarios. Just to show you an example, we carried out just a couple of scenarios trying to use this relationship. We know that invasive species, when arrive in a certain place, the number of new the number of the colonizers is important to determine the population dynamics. And then the initial density is probably a very important driving force for the probability to invade a certain place. And then we tried to understand if we can use just the mechanistic core of this framework to understand if 60 colonizers or 600 colonizers are different in reaching the stability of population, because we know which is the current capacity with the K of the population. We know is about 25 million of animals, and then reached about after about 20 years. And then we tested the role of founder nucleosides on population stability. But we know also if we stress animals, since this is a mechanistic framework, probably we are trying to, we just can obtain information about the number of eggs under under a certain stress, the action of a certain stressor. And then we carried out a lot of different months of acidification experiments, and we saw that the maintenance cost increased in our organisms. And then we studied the effect of the acidification just on the fitness, and we studied which is this effect, which is this effect just on population stability. This is just a very quick result. This is our measured key carrying capacity. This is the scenario 1A, and we can saw that the population after 40, sorry for the Italian labels, 40 months reached the or at low density, after 160 time step, reached the population stability. This piece of time is very important because it's at low density, and then we can probably infer in this case that in this period all factors, stochastic factors, predation and biotic interaction can act in this period, and then can have an effect on the future and the fate of the population. This is a very important moment in during the colonization. When we run models with 600 colonizers, we see that the stability is reached before, is reached before, and this period at low density is probably smaller. The probably most interesting results is when we use a sensitivity analysis, and when you use this a framework, just reducing a bit or increasing, a bit the amount of somatic costs, which mechanistically we can, through our model, to project in the number or amount of eggs produced per lifespan, and then using the acidification experimental results, we can have these results. If we have an ocean, acidified ocean, this population, in theory, never will reach the current capacity, and apart from this spike, just in scenario number 2b. This period is longer in both scenarios, and then potential effects, however, potential effects at stochastic effect, biotic effects at low density clearly can have more room. This is the last slide, and this is the results, just a preliminary results from the biodiversity studies. And then we put the density of brachydontes on this axis, and here this is the Margalef of the associated community. This is a very well-known curve, because we know that the diversity increase with the density of ecosystem engineers, but are arriving at a certain threshold due to some negative factors or limiting factors. The diversity drops. And then, if this is just an inference, we can use this information just to infer on the effect, potential effect of environmental change just on our ecosystem engineers. And if we lose an ecosystem engineers, or more generally, if we have the environmental change or the climate change affecting the density of ecosystem engineers, we can have just this effect. If we can predict the number and the density in our population through this framework, I think that we, in theory, can have more power, and more realism, just to predict the environmental change effect on ecological response. Thus, the idea is to, since it is not possible to use just a mechanistic approach for all steps, we propose just to mix all. And then, just since the ecology is not a rocket, like said Gray-Williams last night, but is very complex. We need to mix, combine a lot of different things, different methods, different views, because we are in front of, we have just a very complex systems in which the variance, variability is probably the most important thing to be controlled. In this is important the oxygen consumption rate, and the respiratory science is crucial in this part, like all experimental procedures, just to get, just to complete this puzzle. I want to thank Brian to have invited me. Just all people here, and then Ellie, Jess, and Francis, and other people from the Brian Helmut lab. These also friends of mine here present. And then this is this research is funded on different steps by different founders, not colonizers. Okay, thank you.